These are Billy's parents. This is Billy. Billy has a problem. It seems as if he has fainted. Oh no! Is he dead?
Welp, time for round 2.
2,000 years later.
Billy, watch out! I'm gonna tag you!
Actually, no you're not.
Oh no! Billy, are you okay? What happened?
Looks like Billy has fainted again. I wonder why.
12 seconds later.
Hi Mom. Hi Dad. Why do I keep fainting all the time?
It's nothing serious. And it's too complicated to explain. And I'm too lazy.
How about you go ask that one goat geek that just came into town?
Okay, I'll ask him tomorrow. I'm just a wee little goat. I can't die. What's wrong with me?
The next day.
Bye Mom. Bye Dad. I guess I'll just stop by the orphanage because you guys don't care enough to tell me what's wrong with me.
Have fun! Billy has fainted. Again.
You need the doctor.
Billy has fainted. Again.
Is he still alive?
I'm okay. I just faint sometimes, and I can't move my legs or body. Okay.
Well, aren't you gonna follow me to the doctor?
Wait a second. Why are we here?
I'm Dr. Geek. What's wrong with you?
I don't know. I was hoping you could help me.
Interesting.
I keep fainting all the time, especially when I get scared.
I wonder why. I already know.
Well, I don't actually faint. My legs, they just go really stiff, and I can't move for a few seconds. It always makes everyone really worried, and they always think I die.
Oh, well, that's because your muscles don't always relax when you're running. You have a mutation.
What? What's a mutation? And how does he keep doing that? The doctor said I had a mutation. What's a mutation? Am I gonna die?
Well, duh.
Ugh, that useless doctor. Of course not, sweetie. A mutation just means there is a change in your DNA. Your DNA doesn't make the gene CLCN1. This means that your body doesn't make the protein CIC1.
Huh?
Okay. Let's start from the beginning. Enter flashback. Oh no! Is he dead?
Welp, time for round 2.
Where did you come from? Unfortunately, he's not dead. He just has a mutation called myotonia congenital. What does that mean? Beep, beep, beep. What are you doing? Reversing.
I like this guy.
Okay, so all living things have DNA, right? It's what makes the proteins that make everything work. Every organism has different DNA, but there are similarities between a kid and their parents, or even with the rest of its species. This is because DNA is divided into subunits called genes. Each gene is responsible for making a protein that the cells need. But in order for the cell to make the proteins, they need to make a copy of the DNA. This is done in the... What? That doesn't make any sense. Okay. Why are we in a restaurant? Don't worry about it.
OMG! They have crabgrass.
Try thinking of it as a restaurant. In the restaurant, there's a book of recipes for how to make all the food, which stays in the head chef's office, or the nucleus. This is the DNA. To make the food, you need to get the recipe from the office into the kitchen, but you can't take the whole book because someone else might need it or it might get dirty and ruined. So instead, you have to make a copy of the recipe. This is similar to how DNA can't leave the nucleus of the cell because it could get damaged. So to make the recipe, the chefs will have to make a copy of it. This process is called transcription. Now, follow me to the kitchen. To make the recipe, you need to make a copy of it and bring it back to the workstation. So, you would open the book to the recipe you need and scan it on a copier machine. Okay, but how does that work for DNA? Hey, what was that for?
Oh look, a diagram.
In terms of DNA, the food is the protein that your cells need to make and the gene is the recipe for how to make it. During transcription, there are two enzymes that are used to help. The first one is helicase, which opens the book or splits apart the strands of DNA. The second one is RNA polymerase, which copies the recipe or the gene. Okay, but how does it know what to copy? Because it follows the complementary base pairing, which basically says that adenine can only bond with thymine and cytosine can only bond with guanine, or vice versa. So what the RNA polymerase does is take extra RNA nucleotides and lines them up to create messenger RNA, or mRNA. For example, if there was a cytosine nucleotide on the DNA, the mRNA would have a guanine nucleotide, and if the DNA had an adenine nucleotide, then the mRNA would have a...
Oh, I know. It would have thymine, right?
Wrong. RNA can't have thymine nucleotides, so it uses uracil instead.
Oh.
So after all the RNA nucleotides are lined up and the mRNA has copied the entire gene or the entire recipe, it can be taken to the ribosome or workstation. Alright, so then what does it do in the ribosome? Translation. Meaning, it makes the proteins. And how do they do that? Well, the mRNA goes to a ribosome where proteins are synthesized. In the ribosome, the mRNA will tell a tRNA what kind of amino acid it needs based on its codon, a group of 3 nucleotides on the mRNA. The tRNA knows what kind of amino acid it needs because it has an anticodon, which is opposite of the codon on the mRNA, though through complementary base pairing. So the tRNA brings the right amino acid and lines it up with the mRNA. The mRNA will always start with a methionine, which is a start codon. Then we'll do the same thing for the next codon, but this time the 2 amino acids will bond together, forming a peptide bond and starting up with the protein chain. Once the entire protein is formed, the last codon will be a stop codon, and the tRNA will stop. Then the protein will go wherever it's needed.
What?
Ugh. Okay, so think of the ribosome like a workstation for the chefs. Once they have a copy of the recipe, they need to bring it to their workstation to actually make the food. And to make the food, they need to get ingredients. So in this case, the amino acids are the ingredients to make the proteins. To get these ingredients, a transport RNA, or tRNA, will bring over the correct amino acid and line it up with the mRNA. How does it know which amino acid to bring? Hey, stop that.
Oh look, another diagram.
Well, the mRNA is split up into small groups called codons. One codon has 3 nucleotides and calls for a specific amino acid. Now each tRNA has an anticodon, which is the opposite of the codon through complementary base pairing. So the tRNA will bring the needed amino acid to the ribosome. The first amino acid brought over is methionine, which is also known as the start codon. Once the second amino acid is brought to the ribosome, it will be connected to the methionine with peptide bonds to create a protein chain. This is like mixing the ingredients to make the food. More and more amino acids will be added until the ribosome gets to the end of the mRNA. Then the mRNA will have one final codon that calls for a stop amino acid. Or this could be like putting the recipe in the oven. This signifies the end of the protein, which means it can go wherever it is needed in the cell. Oh, well, thank you for the spiel, Doctor, but how do proteins exactly help us? Well, Billy's mutation may...
Huh? What's that?
A mutation, Dad, is when there are changes in the DNA that affect the genetic information of an organism. During translation, Billy's DNA sequence was altered, causing him to possess traits that a healthy goat wouldn't have. This means that the nucleotides that are a part of the mRNA sequence must have been changed. We call this a point mutation. Now, there are different types of mutations that fall under this umbrella, such as substitution, which is what... Out of all the point mutations your son has, deletion, you guys want to guess what happens here?
Ooh, wait, hold on, I got it. Is it where a nitrogenous base is inserted?
It's when a nitrogenous base is deleted, sweetie.
I thought it was gonna be one of those trick questions. My bad.
Well, you clearly disappointed me. It really wasn't.
They ask you how you are, you just have to say that you're fine when you're not really fine, but you just can't get into it because they would never understand.
This is also a missense mutation, because the amino acid that was originally supposed to be brought to the ribosome is now a completely different one because of a deletion. The mutated gene sequence now is shifted, in this case to the left because of the missing base pairs. Because the reading frame of this genetic message is now incorrect, a protein is left in confusion and can't perform its job. Any questions?
I don't get this at all, bro.
Will you do me the honors?
Oh no.
I would love to. What your son has here is myotonia congenital, an inherited mutation that causes the muscles in his body to stiffen up after they contract, like when he runs. As a goat, he's going to experience tetanic or sustained muscle contractions, specifically when he's startled or excited. You might have heard of fainting goats, but this isn't accurate. As you can see from my amazing demonstration, Billy is still awake during this and is just temporarily paralyzed. Fortunately, the duration of this is only 10 to 15 seconds. It could be worse, but after he'll just get up and awkwardly walk away. I really appreciate your continued yapping efforts, Doctor, but may I please ask if we ever get to the part where the protein is involved? The mutation that Billy has stops the CLCN1 gene, which makes the CIC1 protein, aka the chloride channel, from forming. In a healthy goat, it's usually found in your skeletal muscles, which help you move. Behind every movement made, these muscles go through contractions and relaxations, which are controlled by certain ions. Now, we get into the part where this protein matters. This protein is in charge of the flow of chloride ions in and out of the muscle cells, which lets goats walk, run, jump, do jumping jacks, front flips. You get the idea. Wow, this protein sure sounds amazing. Oh, right. The CIC1 channels work in pairs with their identical twin subunits. They work together to regulate the flow. Another thing to remember that because of the fact that Billy lacks this protein, you will also make him a bit big on the bigger side.
You mean chunky?
That's one way to put it, yes. Because myotonia congenital robs your son of the chloride channel protein affecting the normal muscle doings, his muscles will undergo sustained muscle contractions, tensing and stretching his muscles. Billy will end up having more muscle mass compared to everything else.
OMG, honey, this will mean that little dude's gonna look like he's been grinding at the gym from birth.
Oh, poor Billy. End of flashback.
Guys, I finally found out what's wrong with me.
OMG, that's great. What is it?
He has a mutation.
Billy, I told you not to faint.
Guys, I'm still here. Just upside down.